mechanisms and methods of tissue fixation. So in regard to basic mechanisms involved in fixation, we have two basic mechanisms. One is additive fixation. Number two, we have non-additive fixation. Additive fixation involves the chemical bonding of fixative molecules to tissue components, in which there is formation of cross linkages or complexes that stabilize the tissue structures. So the fixative chemically interacts with tissue proteins, nucleic acids, or lipids becoming part of the tissue. Classical examples of fixatives that fall under additive fixation include formaldehyde, glutaraldehyde, and osmium tetroxide. So, in a nutshell, these fixatives primarily work by cross-linking proteins, stabilizing the tissue structure and preserving cellular morphology. So the bottom line is the fixative molecules become part of the tissue structure. Cross-linking strengthens tissue integrity and preserves molecular details. Basically, this additive fixation is commonly used for detailed structural preservation, for example, electron microscopy. The second mechanism is non-additive fixation. So in non-additive fixation, there isn't bonding of fixatives to tissue components. Instead, the fixative act by precipitating or coagulating proteins and other biomolecules. So basically, the fixatives disrupt the hydrogen bonds, hydrophobic interactions, or ionic bonds, leading to protein coagulation. Classical examples include alcohols like ethanol, methanol, and acetone. So the non-additive fixatives primarily work by dehydrating tissues, resulting in protein denaturation and structural stabilization. So the fixative does not chemically integrate with the tissue. Coagulation alters the natural conformation of biomolecules. So the non-additive fixation is commonly used for rapid fixation or for specific staining methods like cytology or enzyme histochemistry. When you look at the methods of fixation, fixation of tissues can be achieved by either physical or chemical means. Physical means or physical methods include one, heating, whereby it involves using heat to denature proteins and solidify cellular components. This method is rarely used for tissue specimens, but it's commonly applicable for smear preparation for microbiological studies, for example, bacterial smears. So, in heating, proteins coagulate and adhere to the slide, preserving the morphology of microorganisms. However, heat can cause tissue distortion, and it is therefore unsuitable for detailed histological or cytological studies. Number two method under physical method is microwaving. Microwaving involves the use of microwave ovens to accelerate the fixation process by heating tissues while submerged in a fixative solution. So this process enhances fixation and reduces fixation time for paraffin embedding. Therefore, this enhances fixation speed and reduces fixation time for paraffin embedding. So under microwaving, heat improves the penetration of fixatives and cross-linking of proteins without damaging the tissue. Number 3, we have cryopreservation. Under cryopreservation, it involves rapidly freezing tissue to prevent molecular movement and preserve its native state. So cryopreservation, we can also call it cryofixation or freeze drying. So it's commonly used in histochemistry or immunofluorescence and electron microscopy to study enzyme activity and antigen-antibody interaction. Cryopreservation, freezing immobilizes biological processes and prevents autolysis. So freeze drying removes water, hence preserving tissue structures. However, it requires specialized equipment, for example, a cryostat, and careful handling to prevent ice crystal formation, which can distort tissue. The second chemical fixation methods. One of the chemical fixation methods is immersion fixation. Immersion fixation, the specimen is directly immersed in the fixative solution, allowing the fixative to diffuse into the tissue. So the fixative stabilizes proteins, nucleic acids, and lipids by forming cross-links or precipitating them. Immersion fixation is suitable for small tissue samples or biopsy specimens. However, large tissues may require longer fixation time due to slow penetration. Number 2 is perfusion fixation. With perfusion fixation, the fixative is introduced through the vesicular system, ensuring rapid and uniform distribution throughout the organ. It's mostly applicable to whole organs, e.g. brain and lungs, in experimental studies. So the fixative is injected via blood vessels, preserving tissues in situ. This prevents ischemic artifacts and ensures better preservation of fine tissue structures.